Okay, so thank you everyone for this opportunity, and thanks to Barry and the team at Edelweiss Connect for organizing this webinar. My name is Samik. I am the Chief Technology Officer of SBX Corporation here in Tokyo, Japan. And I want to spend the next 15 to 20 minutes talking about our work on creating a platform which can serve as an engine of scientific discovery with a focus on connected analytics, empowering biology, and with potential applications beyond biology. So I'll probably try to keep my presentation for 15 minutes. And if time is available, I want to spend the last 5 minutes on showcasing a demo video of one of the connected analytics projects which we are doing in collaboration between SBX and Edelweiss. So for the sake of the presentation, I'm not going into presenter mode. I hope that would be okay. So why do we want to create an engine of scientific discovery? So I want to start with the vision which drives our group in general, which is going towards a convergence of human and machine intelligence. And we have a audacious goal which was uh, laid out by our president, Dr. Hiroaki Kitano, in a paper in AI magazine a couple of years back, where he proposed that what would it take to for AI to win the Nobel Prize in medicine. And he prefaced that with the subtitle of creating the engine of scientific discovery. So before I go into the platforms which we have created towards achieving this goal, I want to give a small background of our group. So the group essentially is a team of scientists, engineers, researchers from various diverse backgrounds. We operate under three entities. The first, which you see on the left, is SBI, which is the Systems Biology Institute, a private non-profit research institute which was established 20 years ago, pioneered the field of system science and thinking in medicine and healthcare, and works today at the intersection of complex systems, AI, and computation. SBX, which is the execution arm as we like to think about it, was spun out of SBI to provide the platform services primarily in the verticals of pharma and healthcare, providing consultancy services in the business of biology and translating the research into reality. And as I mentioned at the start of my talk, we envisage the platform to serve as an engine in biology and beyond biology. The rightmost company, which is called SBX Technologies, was spun out of SBX around 5 years ago with a focus on providing these platform and engineering services in areas beyond biology. So for today's talk, I will not be focusing on the work of SBX Technologies, but mainly on the research and innovation around creating platforms to enable scientific discovery out of SBI, and the services and use cases which SBX has been executing in translating the research into reality. As I mentioned, we come from a very diverse background of people, uh, from backgrounds in robotics, in AI, in systems biology. We have worked a lot in establishing standards, both in the field of representation of knowledge as well as in modeling and simulation in the field of computational systems biology, as well as have worked on AI and robotics for a very long time. As you can imagine, to have a vision to create an engine of scientific discovery cannot be done by a single entity or a group of people. So what empowers us is in reality the innovation network and the ecosystem around our platform technologies, which we have fostered. What you see here is a snapshot of our innovation network. The left hand side of the slide primarily is talking about our partners in academia and research institutions, while the right hand side talks about the various industry partners who have been working with us. And using our platforms in various use cases. So as I mentioned, what we do at the core is AI for science, and you can see on the circle various red colored smaller circles, which represents areas where we have our research themes, starting from computational platforms to fundamental exploratory research in systems biology, to applications of machine learning and big data in healthcare. And we have a major focus on knowledge aggregation, inference, and hypothesis generation within the larger context of AI for science. There are two key platform technologies that we have built over the past 10 years to enable both our research as well as to empower our innovation network partners for their own research activities. On the left hand side is the Garuda Platform, which is a connectivity and automation framework which serves as a base framework to connect different workflows involving data analytic tools working together. On the right hand side is our AI and machine learning framework, which is called Gandhara. It includes and encompasses various classes of machine learning algorithms, which are applied on different problems, which I will talk about briefly. One key area which I want to highlight and which has been a major part of our work is around knowledge, cloud, and text mining. So we have within the Gandhara framework a text mining and analytics platform called Taxila, which itself is a platform focusing on the dimension of text. So text, as we all know, is an important dimension in science. We work with we write a lot of papers, we present a lot of our work in text formats, and the ability to actually build a knowledge engine and harness that is an important part of our endeavor. So we have a dedicated platform within the framework of Gandhara called Taxila, focusing on that. I want to briefly tell you a little bit about what is Garuda. So as I mentioned, Garuda is a connectivity platform. The goal of Garuda is simply to enable different tools, data, services to be connected, to be discovered, and navigated in workflows with applications in healthcare and beyond. So when we started the project roughly around 8 years ago, we laid out the thesis of the need of an integrated platform rather than building yet another set of tools to help understand complex data in biology. And we posited it in two papers, as you can see here. A, the need for such an integrated platform, and B, and probably more importantly, the need for a partner ecosystem, what we called the social engineering aspect for doing virtual big science in systems biology and beyond. The Gandhara framework, as I mentioned, is not per se a framework, a specific software or a module for machine learning, but it serves as an ensemble learning platform which integrates a large number of machine learning algorithms for doing analytics, specifically focused within the domains of biomedicine and healthcare. So what we have done is there is a huge plethora of algorithms, different classes of machine learning algorithms. So we have integrated a whole large number of these algorithms on various classes, which you can see elucidated here, and take a wisdom of the crowd approach, which allows us to actually combine these different classes for a given problem and enable us to build specific solutions, which are then connected on the Garuda platform and allows users to operate specific workflows for their needs on top of that platform. So the portfolio of algorithms within Gandhara are very diverse and have been applied in various domains, from side effect prediction to target and non-target classes, prediction of toxicity levels, or drug synergies are some of the classes of algorithms which have been applied within that portfolio. In terms of the platform commercialization, Garuda itself is available as a turnkey solution, on top of which various custom solutions and consultancy services are provided. For example, Taxila, as I mentioned, was the text to knowledge engine. That's a custom solution which is provided on top of the connectivity framework of Garuda and enables specific tailor-made workflows in various domains of use. So I'll skip the slide on Taxila, but I will share the slide deck, and as it has been recorded, you will have a chance to see how Taxila as a knowledge aggregation engine has been built and customized in various domains, such as in drug discovery, in toxicology, and also in areas beyond biology. So in order to drive how we operate by using Garuda as the connectivity platform and custom solutions on top of Garuda based on our AI framework of Gandhara, I want to give one use case and then I'll show you a few of the other use cases. Is a collaboration between Shimadzu, which is one of the major mass spectrometry equipment manufacturers globally, uh, top 3 manufacturers of mass spectrometry equipments. We have a collaboration and a powered by Garuda solution with Shimadzu. So Shimadzu has a lot of mass spec equipments and it comes with its own softwares. Now those softwares process the raw data and allow people to export the data, and then typically they would need to transfer the data from the uh, computer which comes with the device. They have to physically move the files to other machines where they do downstream analysis. So as part of this collaboration, we identified a specific workflow and we built various software modules on Garuda, which are called gadgets on Garuda. So you can see each of these, like Vis Cloud, Gene Annotator, iPath, are different gadgets available on the Garuda platform. And the blue line shows the connectivity of the workflow, which was automated on top of the Garuda platform. So this is now available both as a free version as well as a commercial version, and branded as a Shimadzu multi-omics analysis pack, which allows people to install this pack on the computer where they want to run the analysis using the Shimadzu equipments. It comes pre-installed with the Shimadzu data explorer gadgets built by Shimadzu Corporation, and automatically has these connected workflows, which on Garuda are called recipes, and allow people to run these workflows and generate visualization and analytics in a pre-compiled manner using the Garuda platform and the set of solution gadgets or modules which have been built. So this is one example of the many use cases of collaboration both with industry and academic partners. So I list here a few of the healthcare applications, as well as for this audience, I uh, highlighted a few use cases which are pertinent to toxicology and to the broader scheme of a safer world by design. So there has been use cases on actually AOP driven modeling and simulation for alternative approaches to toxicology. We have built gadgets and pipelines to apply deep neural network based analysis of omics data related to toxicology. The package being called Detox. There are other areas on where we have worked with cosmetic companies and build deep models to help classify different skin types in cosmetics. We have a big project on safety assessment on AI guided risk assessment for compound genotoxicity with a major pharma here in Japan, as well as we are working, as you can see here, with Open Talks and the initiative from Edelweiss Connect to build connected platforms to drive data and models in toxicology. So I just highlighted, for the sake of this audience, some of the use cases which are more pertinent to toxicology and has been a combination of Garuda AI and machine learning models built on Taxila, as well as use cases which has a text mining component of them. So for the sake of time, I'll very briefly go through a few of the use cases. I will make them available to the audience if they are interested. So as I had mentioned, one of the use cases was actually AOP driven modeling and simulation for alternative approaches for toxicology. It was a mechanistic simulation model on hepatotoxicology to predict cell death dynamics, particularly under mitochondrial dysfunction, upon long term exposure to various raw materials and drugs. This was a collaboration, as you can see, was with a major cosmetics company, and we have jointly had poster presentations uh, on this work. The other work which I believe can be pertinent to this audience is the work which we have done on building an AI based intelligent QSAR analysis and decision framework to enable genotoxicity assessment based on ICH M7 guidelines. So, this is fundamentally the ability to actually analyze the text which typically comes in reports from various databases and in silico prediction systems. So you can see here in the green bubble, fundamentally PDF reports are generated. So this is a powered by Taxila solution, and we build different modules, or as you remember from the Shimadzu example, what we call gadgets, to actually extract the text, analyze the text, summarize the text, and produce consolidated AMS prediction scores for different compounds for their genotoxicity. Uh, for the sake of time, I'll skip uh, some of the other use cases on deep neural networks, the detox framework, which essentially developed a deep learning model to identify informative genes from images or three-dimensional plots of actually dose, time, and mm, the concentration of various transcripts under those effects, which was a collaboration which we are doing with the National Institute of Toxicology here in Japan. And one slide which I actually want to end with, and maybe use 2 minutes of my time to show a video associated with it, is the work that we are doing with Edelweiss Connect on open talks and building connected workflows. The concept is very similar to what I showed for Shimadzu. Open talks provides a wonderful platform for access to different types of data, and there is a need to actually enable downstream analysis, be it either for enrichment, for both in terms of pathway enrichment or for functional enrichment, and subsequently identify the set of enriched genes, find pathways associated with them, and being able to visualize those pathways and make mechanistic hypotheses, which can then be translated into new experimental protocols and therefore allow us to explore different hypotheses. So this was a project we did with Edelweiss Connect and the OpenTalks team of getting different data sets as gadgets on Garuda and connecting them with downstream analytics gadgets to build specific workflows. So Barry, please let me know if I'm running over time, but this is just a video which I want to end on. In terms of the open talks workflow. So what you see here is the Garuda dashboard. Each of the small rectangles that you see here are different modules which are called Garuda gadgets. They can be databases, they can be analytics modules, they can be visualization modules, and all of them are interconnected to allow users to build specific workflows or recipes. So what you're seeing in this video is the workflow around the OpenTalks gadget. So we went into the OpenTalks channel. It has two gadgets on this channel where we open one of the gadgets, which essentially gets data from open TG gates. We gave specific conditions, got the samples, and then we send those samples to another gadget, which is called the Open TG Gates Data Fetcher gadget. So it receives the list of samples. So you can see here, it's receiving the sample list from the first gadget, and then it is actually querying mm, the OpenTGG's data fetcher gadget to get all the data associated with the samples which were selected in the first gadget, accessing the data and making it available on the OpenTGG's data fetcher gadget. So if you can see the screen, it has downloaded that data and is showing the various columns with the probe IDs, the gene names, and the conditions under which we had given, for example, valproic acid under high dose for a specific tissue type. And it got all the data. Then what, again, now I want to go to the next step, which is to do the analysis with this data. So it's showing you the data it has downloaded, and then you see it's selecting the set of genes, and it's actually it's selecting the set of genes, and it's sending that selected set of genes to a downstream enrichment gadget. So it's a gadget called Aryabhata. It is an enrichment gene enrichment analysis gadget. The list of genes were sent to this gadget. It allows the user to actually run very specific enrichment analysis workflows. So you can see here, it's actually allowing the user to choose the p-value cutoff, the enrichment type, the database on which it wants to be enriched, and runs the enrichment analysis and presents the results of the enrichment, both in terms of an enrichment bar plot, an enrichment heat map, and the raw data, which can then be exported. So the final part of this video, what it does is it actually takes that set of enriched values, and it was a pathway enrichment which was done on this particular gadget, and then allows users to actually visualize those enriched gadget by opening the files, either cake files, or in this case it was an enrichment on the Panther database, and therefore use another gadget which is called Cell Designer, which allows us to visualize that pathway. So we started with selecting a specific set of samples for a specific condition, extracted the data from another gadget, the OpenTGG's gadget. We got the set of genes. We send it to an enrichment gadget. We did a pathway enrichment under certain conditions. We got the list of enriched pathways. And finally, we downloaded some of the pathways of interest and visualized them on the last gadget in this workflow, which was the Cell Designer gadget. So this is one of the workflows which we have built with in collaboration between OpenTalks and the Garuda platform, as an example of how these platforms can enable connected analytics towards creating an engine of scientific discovery. So I want to stop here and thank you all again for the time and the opportunity.